Hello, I'm Xiaoyue from the Drone Lab at Carnegie Mellon. My talk today is on adapting language models to explore the multidomain protein universe. So what are multidomain proteins? These are mosaics of sequence segments that encode functional or structural units called domains. Domains are found in otherwise unrelated proteins, and they can fold independently in many different contexts. And in this example, protein MPP6, there are 5 domains, right? And they each fold independently here. And the domain architecture of a multi-domain protein is just a sequence of domains in this protein from N to C terminal order. So for example, the domain architecture of the MPP6 protein is L27, L27, PDZ, SH3, and then GUC. And the domain architecture of multi-domain protein is intimately related to its function. So for example, this cask protein, these 4 domains of the cask protein, they each bind to different things. And so that makes the cask protein a good scaffolding protein that can group these different proteins together at the cell junction. And so this modular organization of multi-domain proteins supports rapid evolutionary exploration of new protein functions. And the 2 proteins that I just showed you both come from this huge McGurk family, and this is just one multi-domain family with variable domain architectures. And in fact, there are all kinds of different multi-domain families with different architectures that grant them different functions. And so the domain architecture of a multi-domain protein is a high-level blueprint of its function. And this domain architecture abstraction has been used by many studies studying protein function and evolution. And what we want is a framework that allows us to ask questions about sets of domain architectures in a unified setting. We want a framework that supports exploratory analysis of the universe of domain architectures, comparison of multi-domain complements across taxonomic lineages, and testing hypotheses about domain architecture evolution. And so this is how I'm going to do this. The notion is to represent each domain architecture as a point in a high-dimensional space. And so I'm going to do this in 3 steps. And to do this, I'm going to use this metaphor. So domain architectures, they are sequences of domains, and documents or sentences are also sequences of words. And so I thought, why don't we use information retrieval or natural language processing techniques to put our domain architectures into high-dimensional space, right? Because what we want is we want domains that share similar biological properties to be close to each other in their embedding space. And we want domain architectures that share properties to be close in the embedding space. And that is exactly what NLP and IR methods want, right? They want words and sentences that share similar meanings to be close in their embedding space. And so, right, to do this, I'm going to do 3 steps. I'm going to tell you about these 3 steps in this talk, and I'm going to show you 2 examples of applications of this framework. So the first stage is domain embedding. We represent each domain D as an m-dimensional vector in a m-dimensional space, right? The dimensionality depends on the specific information retrieval or natural language processing method that we use to construct domain embeddings. And in this talk, I show you 2 examples. One is TF-IDF, term frequency, inverse document frequency, and this takes into account abundance of domains in domain architectures, and also whether they are common or rare domains. And also another technique that's called word-to-vec, and this tries to predict a domain based on its context. Right, so in the second stage, we construct domain architecture embeddings, meaning that we represent each domain architecture as a vector in a k-dimensional space. And this can be calculated from the embeddings of its constituent domains. And in this work, I sum constituent domains. So for example, the embedding of A sub 1 is just 2 triangles plus 1 rectangle plus oval plus 1 trapezoid. And once you have your domain architectures in this high-dimensional space, you could do all kinds of different comparative analysis. For example, you could do dimensionality reduction for visualization using techniques such as t-SNE. And so I'm going to show you one example of visualization for exploratory analysis. So here we embedded all unique human domain architectures, right? And we visualize in 3D with t-SNE, and we highlight these architectures from the Magog family in purple. And you can see that these Magog family members are all very close to each other in this embedding space. So that's for visualization. And if you want to quantify the relationship between domain architectures in this space, you can use pairwise distances such as Euclidean or cosine distances. And you can also expand the pairwise distances to setwise distances, right, with the Earth mover's distance. And so this gives you a number between two sets of domain architectures. You can ask, so whether is my set A of domain architectures close as a whole to the set B of domain architectures? And with this, you could compare sets of domain architectures, right? For example, you could compare the multi-domain repertoire in two taxonomic lineages, for example, animals and plants, right? And I am going to show you another example where I compare multiple sets of domain architectures. So I use this framework to validate a simulator, a domain architecture simulator that we published last year. It's called the Markov. I wanted to ask whether the set of simulated domain architectures generated by the Markov recapitulates properties of genuine domain architectures. And we use the set of genuine primate domain architectures as the training corpus and apply our framework. And with visualization with t-SNE in 2D and with two different word embedding techniques, it seems like that the genuine, which are the red dots, and the simulated domain architectures, which are the blue dots, the red and blue dots, they are pretty close to each other in this space. But how close are they? Right, so as a baseline comparison, I also included a set of randomized domain architectures. So I put the set of randomized domain architectures in this embedding space, and they are in yellow. And we can see that in both cases, in general, genuine and simulated domain architectures are in close proximity in this space relative to randomized domain architectures. And so now we can quantify this relationship with the Earth mover's distance. So these are distances among the three sets, right? And as the Earth mover's distance indicates, the set of genuine and simulated domain architectures are indeed closer in this space than they each to the randomized set. So this experiment shows that the Markov recapitulates the properties of genuine domain architectures better than expected by chance. And to conclude, we present a domain architecture embedding framework that supports visual and quantitative analysis of domain architecture data. And this framework has many applications, including exploratory analysis of the universe of domain architectures, comparison of multi-domain complements across taxonomic lineages, and testing hypotheses about domain architecture evolution. And our framework provides a more nuanced representation of domain architectures, right? The relationship between two domain architectures not only depend on common domains, but the contributions of domains that are not shared depends on their distances in the domain embedding space. So I want to thank the Duran Lab and these three people that gave me suggestions and advice, and thank our funding sources, and thank you all for listening.
And we have time for a couple of questions. go back to the slide 19.
24. Oops. Oh, yes.
This example you're showing an example of domain architecture that are easily closed, that you know were closed and are not closed. So I was wondering, so when you expand it, we see all the several other dots.
Mm.
So I was wondering, those other dots, are they also closed? Are they domain architecture that you know close to
These.
the one you're showing?
Oh, yes. So the question was, we have these domain architectures that are from the same family, very close in this space, right, as shown in here. But there are also other dots that are close to these guys. And what are they? Do we know that they are also close, like in architectures to these guys? Yes. So these guys, they mostly contain also this PDZ domain. So I don't have the domains here, but right. So the signature domain of this family is this trapezoid, which is the GOG domain, and I think that's why these guys are. That's what groups these guys together. And also, many of the members of this family contains SH3 or PDZ or yes, SH3 or PDZ domains. And so these could be these are domain architectures that also contain SH3s or PDZs.
Right. So I'm guessing this is the you're analyzing the proteome for what species and for humans, I guess. Would this do you mechanisms work for if you look at any species all at once?
Yeah, so the question was, I showed examples of analyzing human domain architectures, and could this be applied to multiple, to different lineages? I think yes, definitely, and this is exactly why I wanted this framework. I wanted to compare domain architecture repertoires across taxonomic lineages, right? So with that, I think the tricky thing is how do you learn embeddings that are comparable across two lineages? And I'm working on solving that. And once you have that, there are many applications to this. For example, you could identify, say, missing proteins in one taxonomic lineage, right? So maybe there are some domain architectures that we don't see that, but we expect to perform some functions. This could be actually carried out by other domain architectures that are close in this space, right? You could use that to identify such proteins, or you could also use that to, say, annotate proteins with unknown functions. Thank you.
Any other questions? So the question is, first, is the order of the domain important in the domain architecture? And if so, or if not, I don't know, how is it considered in the NLP? Is it considered or not? And what is the importance of that in the comparison between the domain architecture?
So the question was, is order of domains important in domain architectures, and how is that handled by NLP? So yes, domain orders are definitely very important. For in this simulated work, we actually simulated domain architectures that only used domain bigram, so domain combination statistics from the genuine domain architectures, and the simulated domain architectures recapitulate genuine domain architectures very well. So it tells us that the local combination information of domain architectures are very important, right? So how NLP handles that is by the clever designs of their neural network architectures. And here, I am not actually considering order in constructing domain architecture embeddings because I'm summing them. So a permutation of this domain architecture will have the same domain architecture embedding. And so I am actually working on seeing whether you could have different embeddings for, say, a permutation of this domain architecture. and how that affects however they are placed, I guess, in this multidimensional space. But if sequence is too important, if order is too important, then it's not important to model them in your model, because if model is too important, if that is too constrained, then if you have one domain architecture that has exactly these 5 domains, it will have exactly this order. I don't know if that makes sense.